Okay, good afternoon, everybody. This is Jim Pfeifenberger up in Seward, Alaska. I'm part of the Ocean Alaska Science and Learning Center based at Kenai Fjords National Park, but I'm also part of the Pacific Ocean Education Team, or POET, which is an interdisciplinary team of National Park Service staff from both the Pacific West and Alaska regions. And one of our goals in sort of enhancing stewardship of the oceans is to bring communication and education products about, like this webinar series. So we've been sponsoring a webinar series for a couple of years now to try to bring some of the latest in marine research to you, resource managers, interpreters, and other interested folks out there. So if you're interested in some of our past webinars, you can find those on the Ocean Alaska Science and Learning Center's website, www.oceanalaska.org, because we do record all of these and archive them there. I'd also like to acknowledge Sarah Allen from the Pacific West Regional Office. She helps to coordinate this webinar series. And Yvonne Menard at Channel Islands National Park. She is the chairperson of the Pacific Ocean Education Team. As you know, those of you out there listening have probably done webinars before, but you're in listen-only mode. We'll save questions for the end of the webinar, and there's a little question box where you can type those questions in, and then I will read them aloud for our presenter, and we'll take questions at that time. Before we finally turn it over to our presenter today, I'd like to let you know that we're going to have another upcoming webinar next month. The exact time and date is not set in stone yet, so I'll just have to give you a teaser, and you'll have to look for an email giving the exact date and time for that one. But next month's webinar will be Karen Matsumoto from the Seattle Aquarium, and she'll be speaking on sort of a citizen science-related project. She's titling it Culturing Ocean Science, Integrating Traditional Ecological Knowledge and Western Science. So it's going to highlight sort of a partnership program between the Olympic Coast National Marine Sanctuary and the Seattle Aquarium that brings ocean literacy principles to rural and tribal communities, but then also, you know, capitalizes on the traditional ecological knowledge that exists in those communities. Should be a really interesting talk. Look on your email for the exact time and date in late May. Okay, so today we have with us Jessica Miller. She is a professor in fisheries and wildlife at Oregon State University. She's actually based in Newport at the Hatfield Marine Science Center, and she has been involved in a number of the marine debris issues that have involved, you know, live critters floating all the way across the ocean. So she's really been involved in a bunch of that and wants to share with us some of her knowledge about that, some of the latest that she's learned, and I'll turn it over to Jessica at this point.
Thanks, Jim. Good afternoon, everybody. I'll preface this by saying I've done several Adobe Connect seminars, which is very similar to a webinar, but this time there's always been people in front of me, so this is the first time I'm effectively talking to myself in my office. But I think it should all go well, and I'm familiar with that chat box, so if you have serious trouble hearing me or some other issue, feel free to type in there, because I have two monitors and I can see what's going on. So as Jim mentioned, this has been really a very spontaneous, unexpected, obviously, research opportunity that sort of literally landed in our backyard. As you know, I'm a marine ecologist, does a lot of work with fisheries and coastal resources along Oregon, and we were also surprised last June by the arrival of this dock, which you can see here on the screen, which is really just less than 4 miles north of the Science Center here. So a very fortuitous landing of this structure, which added a new dimension to the complexity of the social, economic, and human tragedy associated with the tsunami and the earthquake in 2011. It was quickly and very abundantly clear that there was a tremendous diversity of species and a high abundance of organisms living on this dock. as it arrived. And that really sparked and spawned this research project, which now involves several colleagues around the country, as well as our state parks and our Fisheries and Wildlife Department of the state. So I presented a little bit of this work at the Alaska Marine Science Symposium back in January and met Jim, and he thought it might be of interest to this group that signs into these webinars for the POET series. So I've expanded this a bit to talk about what's happened, because it's been very busy for us in terms of marine debris, Japanese marine debris, in February, March, and now in the beginning of April. So I'm just going to cut to the punchline, and then tell you a little bit about how we've gotten here and what we're seeing to date. So this evolved and evolving research project involves collaborators in California as well as Connecticut. And we're trying to document really the arrival, so the species richness, the relative abundance, and the location, at least subsample some of this biofoul Japanese tsunami marine debris. And this map shows approximate locations of the 49 distinct items that we've sampled, right? So our new, in the world of too many acronyms, we're referring to these as JTMDs, so Japanese Tsunami Marine Debris items, biofouling 1 through 49. It includes a whole suite of objects from buoys, docks, as you've seen, several pangas, boats, as in the lower portion of this figure. Have been arriving all up down the coast. Our sampling, probably due to effort as well as oceanography, has focused in Oregon and Washington, and Hawaii makes up the majority of our samples, but we've recently acquired some samples from BC and a couple from California. So to date, what we've seen on these 49 different debris items is more than 140 species. And by that, we are focusing primarily on coastal biofouling species. A handful of those are common pelagic species, so species that complete their whole life cycle in pelagic waters and typically show up on coastlines around the region on ocean debris. But what's unique about this event is the high species richness and abundance of these species that are really Japanese coastal fouling species, or benthic species. So I thought I would run through a little bit of the history or the chronology of how we've sort of fallen into this research topic, along with some preliminary results of what we're seeing, and give you an idea of the research questions we're trying to tackle. And interspersed here is a suite of some of the species we found on that first dock, including a granular claw crab. This is Dignified Victory, which is fairly common on both sides of the Pacific. But we also see species such as the Asian pink barnacle, Megabalanus rosa, which is what we consider sort of an indicator species. It's been very, very common on the Japanese marine debris, and it's one of the species that helps us quickly determine if a debris item is of Japanese in origin. We've seen chitons, limpets, several different species of bivalves, 2 different species of sea star and an echinoderm, as well as hydroids, and a colleague of ours is working on the algal composition of many of these debris items, which, as you can see from this picture of the dock, especially the ones that are arriving last summer, very, very heavily colonized by different algal species. So as I mentioned, for us it was a real wake-up call starting back in June, so almost a year ago now, when really we just heard, actually in the hallway right here outside my office, one of our colleagues who gets a lot of reports from just odd things people observe on the beach said, gee, something rather large washed up on shore on, this is called Agate Beach. Maybe I should go take a look after work. So that's what we did, and we were pretty startled. You can see this is a 66 by 19 by 7 foot dock, incredibly well made, over $800,000 US to build these docks, and they had only been placed in the harbor, Misawa Harbor in Japan, in 2008, so a little less than 3 years before the tsunami. And you can just see this mass of settlement on all sides of the dock. This Undaria, which is a highly invasive brown alga. Again, that Megabalance rosa, the pink Asian barnacle. This is an example of the reproductive portion of the Undaria. So many of these individuals were reproductive, actively reproducing or bearing eggs in the case of some of the crustaceans. Several different species of barnacles, sponges, bryozoans. The sea star I mentioned, the North Pacific sea star, which is known to be highly invasive and has invaded and caused extensive economic and ecological harm in Australia, was found only inside the hold. So this dock structure has ballast, so two open areas under that hold on both sides, used to help stabilize and level this structure, which was really the prize of the fishing port in Misawa. They were very excited to get these 4 docks placed in 2008. It really dramatically improved their processing capacity. And the only place we found the sea stars and this debris item were inside the hold, and this hold was flooded. So likely these entered as larvae and managed to maintain some low level of growth during their year-plus transit across the Pacific. Anemones, here's a picture of the Middleus gallopinsialis, the blue mussel, common in this portion of Japan, northern Tohoku region. But it is really the sentinel species for our debris. It's on the vast majority of the debris we see always has this species in various numbers and at various reproductive states. And here you can see the pelagic gooseneck barnacle, right? And that's the classic pelagic species we expect to see on debris and that you often see occurring on debris, although we probably have some of the largest individuals associated with some of this debris arriving from Japan that most people remember ever seeing. So the next few slides are just to show you a bit of the expanse, sort of the depth of the breadth of the organisms we saw on these structures. Here's the surface covered with a variety of algae, of which Gail Hansen here at OSU has identified several to be Japanese in origin, and over 6 inches thick of multiple layers of barnacles, mussels, algae, all on all sides of this structure. Real amazing diversity. Over 20 species of polychaetes. Here's a scale worm, again some of the green alga, and just the real immense diversity of and abundance of species that successfully arrived in this dock. Right, so then this led to the discussion of how we sort of educate the public and how we might work on how to prioritize sampling of different debris items. So some of the information we've developed really focuses on how people can understand some of these differences and what kinds of species we're seeing. So in blue here, these the pelagic gooseneck barnacle, an oceanic species that completes its life cycle at sea and commonly arrives on marine debris. We even saw some whale barnacles on the dock, which were fairly fascinating actually to see an item that doesn't normally arrive on our coast, but is a pelagic species. But then there's the suite of coastal biofouling species that, as adults, do not normally cross the Pacific in such numbers on such a diversity of objects. And so that includes several of these species listed here are on various worst invasive species lists. The Undaria and the Northern Pacific Sea Star make a list of 100 worst invaders. And here we put Crassostria gigas, which is it's a cultured species. It's not. It's a resource species, and it's brought into the country for culturing. But what normally happens is that that broodstock of those individuals are quarantined for anywhere from 1 to 4 years to determine their pathogen parasite load before introducing it into the Pacific Northwest, the coastal environment. And now in this case, we have had several debris items with numerous oysters arriving, and we really have no idea of their pathogen or parasite load. So that's a part of what we're also trying to get a handle on. So now that we've been working to identify these 140 plus species and then categorize them at some level so we understand who's which species are native to Oregon or Washington might have settled on these items when they arrived in our coastal waters. So sort of the gross cut for the first level of categorization, it's just useful to think that we have native species, so those would be such, for the West Coast, as our California mussel, species that evolved in a given area or arrived without the intentional or accidental intervention by humans, sort of their natural distribution. There could be non-native species, or species that are in the region. It's attributed to human action that have enabled them to overcome what we at least currently perceive as fundamental barriers of transport or movement. But then we do have cryptogenic species, so species we just can't really tell where they're from and what their transport and dispersal history was. And we certainly have some of those on the tsunami debris. And then some of those non-native species can establish self-sustaining populations within a region outside their native range. And they're often referred to as naturalized or established. And there are numerous naturalized species that don't necessarily have negative impacts to the economy, the environment, or health. So it's really only that last category there, the invasive species, those non-native species that have naturalized and are also having measurable negative impacts. The European green crab and the Japanese oyster drill are good examples on our coast of species that are having impacts on the east coast and the west coast of the U.S. So because there were obviously at least 11 different species of known invasion concern, that's the dock up there in the picture right here, and our state Department of Fish and Wildlife took quick action, and that's Mitch Vance actually blow torching the side of the dock. So their control response was to pretty immediately, so we went out that very next day to this structure, the first debris item that had arrived with extensive biofouling that we know of. We collected a bunch of samples and were determining that there was this suite of non-native and potentially invasive species. So the state went out and scraped the sides of the dock, piled all of that material up on tarps and in bags, dragged it and hauled it above the high tide line, at which point it was buried relatively deep below the sand, an estimated almost 2 tons of biomass. And then it went through to get to nooks and crannies and attempted to, inside these bumpers, to use the blowtorch to actually then kill off any remaining possible species, because the stock was on the beach for another month or so before removal. And what really, I think, surprised all of us was the level of national and even international interest. So in addition to over, I think, 999 different articles and stories, and it's a couple of weeks after we found this dock and sampled it, this is a composite image from OSU's Coastal Imaging Lab, and you can see it's like an anthill. This is the dock right here, and this is all of the different tourists over, I think this is a 24-hour integration. You just see the tremendous level of interest and visitation, as well as the Coast Guard helicopter here flying by, doing a flyby on the weekend. So this site, meaning this part of Oregon State Parks and the beach, typically receives about 2,000 visitors in June. So we got 20,000 visitors in June of 2012. I would meet people that would say things like, I was in Seattle and I thought I really had to swing by and see this dock, even after it was fully cleaned off. So it really grasped some level of fascination by the public for a lot of different reasons, I think. So after much discussion and several bids, the way to remove this structure was decided that it would be cut into 5 pieces by Ballard Diving and Salvage, an operation out of Seattle, Washington. And so you can see here, this is an example of that almost finished one of the first cuts, which proved to take quite a long time. And this is pretty amazing infrastructure and talent these people have at actually dealing with this in a high-energy surf environment. So this dock was then removed, and a chunk of it has now been returned to the Hatfield Marine Science Center and has established as a memorial and an educational display right outside my office here in Newport. And the removal in this case, so cost about $85,000 to remove this dock that cost $800,000 to build, that had successfully survived a tsunami, earthquake, and thousands of kilometers of ocean transport. So then we were faced, meaning John Chapman, a colleague of mine here at LSU, and Gail Hansen, another colleague. We had this suite of species, this evolving phenomenon. An Oregon Sea Grant was generous enough to provide a little seed money. So we took this sort of four-pronged approach to dealing with the debris. We wanted to basically first understand who we had, what species were there. So we've been working with Jim Carlton at Williams College, who's perhaps the leading expert in marine bioinvasions, and he's helped us develop a cadre of taxonomists with different specialties. And we've sent them packages of mollusks and crustaceans, or anemones, and bryozoans, and hydroids to different people that can help us using morphology determine what species we're seeing. And we're also working with the geneticist at Moss Landing, who's helping us then generate genetic barcoding of those individuals in conjunction with those IDs, and increase that amount of available genetic information online. And then we're also trying to better understand, I mean, in some cases, in the case of the dock, obviously, it's a bit of a floating island, and it really appears that some of these organisms will actually sustain themselves off of other organisms living on the dock. And in some cases, some of these species are annuals. So there appears to be some local reproduction, or what some refer to as self-recruitment, occurring. So we're also compiling information on the size and abundance of a variety of the most commonly occurring species, so we can better understand their potential growth histories, potential recruitment histories. And we're also combining that with some chemical and structural analysis. In this case, mostly of the mussels. As I said, that Middleus has been abundant on debris items all across our collection location. And one of the types of research I do a lot of is looking at the chemical composition of hard parts, be it otoliths or bone or shell in organisms. And it can be very informative, particularly when, as in the mussel, there's some sequential and growth formations that you can track. And so we have added that component in an effort to understand something about how fast these mussels were growing during the transit across the Pacific, because one of the items that really surprises many ecologists and biologists is just that so many of these coastal organisms that are used to residing in highly productive areas could arrive in such good condition, reproductive, and apparently surviving quite well, even though they crossed relatively low areas of low productivity in the North Pacific prior to getting here. So this just shows what the dock, and as you may have heard, there's two of these docks, but this is really obviously the largest item we sampled that has the highest species richness. So at this point, and we think we're pretty much done with the morphological species identification component of this first dock, 119 living non-pelagic species, and 11 of those were known to be invasive. Our current assessment is about half of those are not known to occur on the East Pacific coast. That actually is really Eastern Pacific on the west coast of America, right? So species that are already in Oregon, Washington, or California. And it's important to note all of those could be unique populations, so they could have local adaptation or be bringing in different gene variants. And so another piece of the work that John Geller, our colleague, is doing is looking at some of these more abundant species, such as the Middleus, looking at the population level genetic variation associated with the tsunami debris, and comparing that to what we know about the genetic composition of this species in Japan and some of its other locations. And we have a couple colleagues. In this case, Dale Calder was the taxonomist working on us with some hydroids on the first dock, and they've already leapt ahead and actually published a paper here because they were quite excited to find this species of hydroid that we collected on mussels on that first dock, and it's clearly a Japanese species that has arrived. The first evidence for transoceanic dispersal of the species arriving associated with that dock. And to date, we found this hydroid on several, if not most all, of the different debris items we've sampled. And as I mentioned, just a little more detail on the growth reconstructions that we've been doing on the dock, as well as the subset of the other 49 debris items we've sampled. You can see here this yellow arrow indicates the growth axis of this mussel. And then what this graph up here on the left shows you is the amount of barium to calcium. And calcium is considered an internal standard. In this case, this shell is a form of calcium carbonate. So it has sort of a chemically determined amount of calcium, and we can compare the rate of deposition of barium, which in this case tells us something about the water mass that the mussel was residing in, as well as its relative composition of trace nutrients, trace elements and nutrients. And so what we're seeing quite commonly is this region of higher barium, so elevated elemental concentrations in coastal waters, and then what we likely are interpreting as this crossing of the Pacific, if you will, so low nutrient element waters, and then its arrival into our coastal waters prior to beaching itself on Agate Beach. And we're seeing very similar profiles and able to then use some mussels from Japan that were residing in Japan during the tsunami and survived the tsunami to provide some ground truthing of that. And then what we see is a lot of these different debris items have very different species compositions. And we'll be looking at how the species composition and the growth, and we can also reconstruct temperature by looking at oxygen isotopes in these mussels, and really create a dispersal history for each debris item and relate that to how that is related to or influences the species composition on those different items. And similarly, what these data are telling us for the dock is that a high proportion, a large percentage of these mussels, this is just a size frequency distribution of the Mytilus, 583 Mytilus that we've collected off this dock. But they range very small to quite large, several years old in size. And what this chemical signal as well as some other ancillary data are telling us is a large amount of settlement occurred in Japanese coastal waters, but after the tsunami. And about the same time we were sort of narrowing down on this evidence was the same time we started getting debris items such as fishing boats and some terrestrial debris that clearly were not colonized prior to the tsunami, but they were arriving on our coast heavily colonized by Mytilus and other species. So several lines of evidence support the fact that this sort of mass of debris that resided for some period of time off of Japanese coastal waters experienced pretty extensive settlement after the tsunami outside of its sort of original habitat. John Chapman, as I mentioned, a colleague here at OSU, is looking at the amphipods and some of the, in fact, different fly that we found on the surface of the dock and reconstructing their size frequencies and cohorts, looking at getting some idea of how many generations and how much self-recruitment might have been occurring for some of these rapidly maturing species that are also common on several of the debris items. So a lot of times when we talk about this, people say, well, haven't things been drifting over from Japan forever? And why is this different? And it's kind of hard to document exactly what's been drifting over from Japan and what exactly has come on all of those items. Although to say there are several researchers that have spent the better part of 30 or 40 years hunting for adult examples of adult individuals that are adults, fouling species arriving at different debris items. And other than, say, hull fouling, so about occasional individuals on ship hulls, not that occasional, it can be fairly common, but that's probably the currently understood invasion vector that's most similar to the Japanese tsunami marine debris. And I think a couple things that are really different is this little box highlights the largest tsunamis that occurred in the 1900s in Japan. And The next largest, so the Tohoku, the 2011 earthquake generated a 30-meter tsunami wave. And the next largest wave was just a third of that, a 10-meter wave back in 1993. And there's been an extensive coastal development infrastructure in that 20-year period. And these docks are a really good example of that. As I mentioned, these each cost almost $1 million US to put in. And the only other docks like this in the country, in Japan, are in Nagasaki, and were not damaged by the earthquake or the tsunami. But all 4 of these docks were sucked out to sea. And they're a really good example of highly engineered, obviously very seaworthy structures that could support a large living community. So we've had this explosion of synthetic plastics, vinyls, PVC, styrofoam, really well-made fish totes, a whole variety of objects intersecting with one of the largest waves, leading to probably greater energy and potentially injection of this debris into further offshore than some of the smaller waves in prior history might have taken some of those items. So getting them further out into the coastal currents. So as we were working on these different topics, we really realized that a little more coordinated an approach to trying to quantify what could be referred to as the initial stage of the invasion process, right? We don't know of any of these species, and hopefully none of them will invade or become established or naturalized on the West Coast and cause any negative impacts. But normally, once we have an invasive species, it's all forensics and hindcasting. We're figuring out what the vector was and which event was most likely leading to the success of that species. And in this case, we have this large field of debris coming, so we can characterize that arrival stage. And that with colleagues, I mentioned, Jim Carlton, as well as John Geller, the geneticist at Moss Landing, and Greg Ruiz at the Smithsonian. We've teamed up with them to try to do exactly that. So this is from the National Research Council, and it's just sort of a map of the steps of the invasion process from the arrival, so say the inoculation of the novel environment is the first stage. And one of the greatest predictors or indicators of successful establishment of non-native species is something referred to as the propagule pressure. And that's really a synthesis of the species, their number, the abundance arriving, the density, the number of debris items in this case, or ships and ballast in the case of ballast water, and then their quality. Are they reproductive? Are they barely alive? Are they growing? So when you put all of those together and measure this pressure of the inoculation or the arrival, that's one of the best predictors to date of invasion success when people do these retrospective analyses. So we proposed to NSF that we could try to quantify as much of this propagule pressure as it was happening associated with this novel transport vector, the Japanese Tsunami Marine Debris. And so that's really what we're trying to do now. Very similar to the approach I outlined for the dock. We've taken that approach with all of these other 48 debris items. And as many of you may be aware, there was another dock. So those 4 docks in Misawa, one dock arrived in Agate Beach in June. One dock was spotted passing Hawaii in September of 2012. Third dock was found in December of 2012 on the Olympic National Park on the 18th of December. So thanks to National Park Service employees, Steve Kratkin and others, and Alan Place, who works for Washington Department of Fish and Wildlife, we went out to access this second dock. And from a biological standpoint, it was a really interesting comparison. So it was an identical object 6 months later, arriving in the winter instead of the summer, in a very high energy environment. So we were able to sample that. In this case, we were more prepared to take quantitative samples, quadrats on different sides of the dock. But these pictures highlight, one, the incredibly rugged location this was in, so very different to the Agate Beach site. And the high energy, you can see the peduncles of the lepus, the pelagic gooseneck barnacle here. They tied themselves in Gordian knots. It was so high energy. And this somewhat fuzzy photograph on the right of your screen shows how many, these are barnacle scars of not the pelagic barnacle, but coastal barnacles, and a variety of other organisms that have been beaten off, likely by this high energy rocky environment. We found very similar complement of species, the pink barnacle, the middleus, the anemones, that hydroid I mentioned. We also found this commensal hydroid right here lining the inside cavity along the gills of the middleus. And that we've only seen in one other location. So we're exploring what is known about whether that kind of commensal species can infect or live in our local middleus. We're probably raising more questions than we're answering as we're scrambling here to document a lot of this different debris. And just briefly, in this case, it was a somewhat less accessible site than that Agate Beach site, but a very similar scraping and removal of organisms remaining on the dock was followed by a light bleach solution in areas, nooks and crannies to kill any remaining organisms. And in this case, you can see here they're lining up the bumpers which were on the all sides of the top of the dock. And that was quite interesting because when the Agate Beach dock was removed 6 weeks after it landed, even after the burning and scraping, we found living barnacles and mussels inside some of those bumpers. So getting those off this dock was deemed a good idea, or was a good idea. And now this has been also removed from site at a fairly high expense. And so even though this was beaten, many organisms were smashed off before we even accessed it, and it was winter, we still documented 67 coastal species, 4 different pelagic species on that item. And I want to briefly mention the work Gail Hansen's been doing with 2 Japanese colleagues. She's been combining morphology and genetics to identify the marine algae. She focused originally on those 2 docks, but has also been looking at other different items. And she saw a very similar pattern, where she saw about half the number of species on the second dock as the first, but 30 plus algal species on that first one. And similar breakdown in the sense that species already known to have invaded Asia or be Asian species comprise about 50% of those, which is very similar to what we're seeing for the animals. About half of those are cryptogenic, and a small portion, about 8%, are known to currently reside on both sides of the Pacific. And she's also found some very invasive species that are concerned in several locations, the Undaria as well as Codium, dead man's fingers, and Greta lupia. So one of the really interesting aspects of this is the dispersal trajectories. And this is a graphic of the wave height generated from the tsunami and its distribution after a couple hours. So this is just 28 minutes after the tsunami, and this is 2 hours, almost 3 hours later. So you can see a lot of variation in where the debris items went. This is a picture, this map is a flight the Coast Guard did, the Japanese Coast Guard did on March 20th, so about 9 days after the tsunami, where they marked a lot of the different debris items. And this one right here, that dot, that's the 3 docks. So that's 3 of the docks from the Misawa harbor that have been pulled out of port during that tsunami. So it's quite interesting because the biology, in this case the presence of this Megavalens rosa, which is quite common on both docks, in fact, has a more southern distribution in Japan, and it's not common at all where the docks originated. So this is another piece of the evidence that supports the fact that these items, some of them swirled around and traveled south, and then were colonized in some cases by a suite of species with different distributions than where those items were originally from. And just the fact that we have 3 items from the exact same location that end up in drastically different endpoints highlights the stochastic nature of the dispersal And what we're hoping we'll be able to do in part by a lot of these different boats and obviously the docks can be traced back to their point of origin. So we will have some natural tracer studies. Some number of these, say about 25 or 30, we'll be able to say, well, this is where it ended up, this is when it started, and this is the community that was associated with it, and get a better idea of how that colonization over time has happened on these different objects. So I want to expand a little bit to some of the other kinds of items that we've been seeing. Here's an example of these fishing boats, often referred to as pangas, that very, very common in Japanese waters, often pulled off out of the water at night. But personally, here in Oregon and Washington, we had a spate of these, almost one or more every week through February, arriving all up and down the coast in both Oregon and Washington in various states of colonization. And this boat in particular, Dignified Victory is its translation of its name, arrived on March 22nd on Long Beach, Washington, which is the southwestern tip of the state of Washington, 740 days after the tsunami. And this boat was heavily colonized. But it also had 5 tropical fish living here. Well, the idea is, from people that sampled this boat, is that it was submerged, sort of neutrally buoyant, where just the gunnels were above water. And so they envisioned that the fish, water was filling most of the boat, and the fish had access to most of the boat. It was pretty remarkable that 5 of these rock bream Opognathus fasciatus, this reef fish with a very limited western tropical distribution as adults. And the adults live in quite shallow waters, up to about 10 meters. And the adults get really large, about 80 centimeters. So these fish that were in the boat were about 40 to 50 millimeters. The adults are about 800 millimeters. So what this lets us also do is take, this is a picture of the otolith, the lapillar otolith of this fish. And because we know that most otoliths have daily increments, unlike the mussels, which just lay down increments and might have an annulus or a winter mark, we can determine, at least estimate, how many days old these juveniles are. And then now we have another kind of tracer, and we know because of the limited distribution, we can really get a stronger handle on the minimum transit time of some of these objects and further explore how this, again, probably upwards of 50 species successfully survived, and in some cases thrived, this long journey across the Pacific in relatively low nutrient waters. This case we saw scallop, many of the different barnacles, including that Asian pink barnacle. This, in fact, is a Middleus, completely enshrouded in 2 kinds of stalked rhizome and hydroids, several limpets, a different kind of shore crab than we saw on any of the other boats, all still alive in this boat arriving quite a long ways after the tsunami. So to give you an idea of the diversity of what we're sampling now, or really been sampling for about almost a year now, buoys, fairly common, mussels, oysters, barnacles, hydroids, occasionally bryozoans, common on those objects. As I mentioned, a whole suite of different boats. Each one seems to have something unique about it. One broken boat had a fiberglass plate just covered with 2 different species of glass sponge. This is not a great photo, but there was a stalked as well as a base sponge, very small, really less than about, smaller than an inch in this case. And then this is like an opera, a really beautiful bryozoan. That this is actually a picture from a Japanese coastal guide. This like an opera species was all over this fiberglass plate. The only debris item we've ever found it on is that one boat. This large red steel pipe, which came up on the Macaw tribal lands, again in Washington, managed to float over here with some different barnacle species attached to it. We're currently finding wood now, both naturally occurring wood as well as dimensional lumber. that has, in some cases, the megabalanus, that Asian pink barnacle, sometimes high numbers of mussels in this case, and lots of lepus. The wood is proving to be quite a challenge. And when do you call a piece of wood Japanese Tsunami Marine Debris? So at this point, if it has megabalance, it has that pink barnacle, we call it such. If it has mussels, we take some of it, we document it, and we take samples for genetics. And what we're really hoping is Middleist Gala provincialis does occur in California, but it's highly likely there's some unique genetic variation, and we'll be able to distinguish Japanese Middleist Gala provincialis from California. So we might be able to rule out transport from the south versus transport from Japan for some of these debris items, which may also provide us some novel insights into just natural rafting on woody debris. This little boat down here, the Right Prophet, is kind of a fun story because this one arrived in Hawaii back, I believe it was in February, and the family blessed the boat and ate the mussels for dinner, and then reported the boat to different authorities, at which point I don't think there are any samples left to be had on that one. And what we're seeing commonly, really a large pulse, it's been trickling in since February, but currently this mortise and tenon, post and beam construction is very common, even in new construction in Japan, not at all common in the U.S. And there's now a whole variety of information, if anybody's interested, I can forward it to you on how prevalent this is in Japan and how most of these items are most likely Japanese. And this gives you an example of some of the items that we found just off the beach here, and several of them have Middleist on them, and a lot of different hydroids, and some evidence of other flowering species that have been either desiccated or smashed off. And so that's the current pulse of debris we're seeing now. And it's a bit more of a challenge, as I mentioned, to try to quantify or sample, but we're plotting ahead. And so just a couple more slides. Again, I wanted to sort of revisit some of the diversity that we've been seeing on debris items. As I mentioned, two different kinds of shore crab. One that is invasive on the East Coast, only arrived in small numbers on the first debris item, but then we got yet a different coastal species recently on the Long Beach boat, over 700 days after the tsunami event. Several species of urchin, compound and solitary tunicates, a whole suite of mollusks, examples of crabs, coastal crabs that are common and currently present on both sides of the Pacific. Pretty rare find of these tropical reef fish, sort of the sentinel species for Japanese tsunami debris, the Middleist, several species of bryozoans and amphipods, almost 30 different species of worm, and a couple of different anemones. As I mentioned, that barnacle diversity we've been seeing. And so one of the ways and to keep up on this, or to find out what we've been seeing, and actually I could probably just bring this up on my screen, we keep a blog, which I just recently renamed from the Japanese Tsunami Doc blog to Biota on Japanese Tsunami Marine Debris, to more appropriately cover what we're really doing now, which is trying to characterize the biota on a subsample of all that debris. And that as species lists, as we come up with them, there's a collection protocol that we've been sharing with local citizen groups and state employees that are interested in helping us sample or just document what they're seeing. And then we have links to an ongoing digital image database we have of Japanese tsunami marine debris biota, as well as, at this point, the different debris items at various stages of colonization are located on that Flickr page. So those are good ways to find out more or keep up a little bit on what we're seeing and how it might be relevant to your locations as well. And then, so I'd be happy to take any typed questions if there are any out there. But that was about all I had covered this afternoon.
Well, thank you very much, Jessica. It's really fascinating and clearly an ongoing story as it keeps changing as new things wash up. So yeah, folks, if you do have questions, open that question window and type them in, and I will read them, and we'll start off with one here. Jessica, it says, are you finding evidence of any burrowing organisms such as shipworms within the organic woody debris?
Yeah, great question. This is one that, if you know, Jim Carlton is near and dear to his heart. We have just recently, so again, in the last month or so, the woody debris has been arriving. There have been some of these post and beam, as well as a couple of just logs. In these cases, we've tried to get the pallets and the shells out of the wood. We haven't had a lot of luck yet, and we're basically cutting off chunks, letting them dry out, going to start beating them with hammers and getting out the shells, the things that will help us identify who's there and get a better idea, because there are differences in species composition from coast to coast, and some that are known to just really be common in the open ocean environment, and we'd like to figure out if that helps us classify the wood in terms of its point of origin. So not as much as you might expect for some of these pieces of wood, where all evidence suggests they've been in the ocean for a good couple of years, but we are seeing some, and we are trying to track that too.
Okay, thank you. Another question is, could you clarify the sort of range and location of where those live fish would have come from? You said Western tropical. Does that mean somewhere in Japan, or does that mean the boat passed through somewhere further south and then came back up?
Yeah, so the distribution from all we can find of that fish is southern Japan and possibly a little further south. It's rare in the western Hawaiian Islands, the far west portion of that. So it's a relatively limited distribution, and even more limited in terms of where that species is abundant. And it's also a cultured species where they actually supplement some natural populations. So it really looks like all lines of evidence point to the fact that it likely was from the southern Japan region, and that that boat might have drifted, possibly gotten hung up on something, spent some period of time in that region, possibly gone somewhat farther east prior to recirculating and ending up in Long Beach. So it has a relatively limited distribution, and definitely points to that boat spending some time in southern Japan or nearby prior to moving on.
Okay, thank you. Here's another question. What does the information on biota identified tell us about mitigation measures in the future? For example, how would you best eradicate invading biota if you could detect the debris before it reached shore or before it reached land?
The whole question of whether you can actually sample or access any of these before they get to land has been ongoing and interesting, and I've learned a lot about marine law and fluid, especially for large items. There's a lot of complications. So although it was discussed with the docks and other items, other than some Hawaiian folks basically towing those boats in and adopting them when they've been out in the coastal waters, I don't know of any active eradication prior to landing, although it's been discussed. What's usually been the stance, at least when I can speak for Oregon and Washington, is to document, keep their eye on as much as they can, and as soon as they can, get these objects off the beach, out of the high tide zone, or otherwise cleared of any biofouling. And I know in Oregon that's been happening sometimes within 2 hours of a sighting, because they've got contracts with folks around the state. So they see a boat, they make a call, within an hour or two, there's a bobcat or whatever needs to be there, and they're hauling it up and taking it to some disposal site. I'm not sure if that answered your question, or if you were getting more at once things are already reproducing here locally. But that's sort of the reality of what's happening on the ground in Oregon and Washington.
Okay, here's another sort of related question, and that is, is anyone looking for or monitoring, for successful recruitment of these flora and fauna in the natural habitats near where the debris washed up?
Yeah, it's a great question. And right now, we certainly aren't, because we're all so busy. And what we're trying to do is work with Oregon Sea Grant, and we're currently actually writing a proposal for this, but to work with Sam Chan, who's the Invasive Species Coordinator for Sea Grant in Oregon, and combine exactly that. So we now know what Japanese coastal fouling species arrived in the debris. And what we'd like to do is take a suite of coastal Japanese fouling species that did not arrive in the debris and really develop a information network of different life history traits, known distributions, temperature tolerance, salinity tolerance, and develop a bit of a ranking. So based on our estimation of abundance, we know where at least the items we've sampled have been sampled from. So we have an idea of the propagule pressure. That's what we're trying to quantify. So we should be well poised if we can combine those data with information on life history traits and what else is known about these organisms to develop at least a prioritization framework, a ranking with information on what species to look for, what their larval or early stages appear to be, and work with those in the region that have the then ability or the potential to add some monitoring locations to help assess some of the potential impact and hopefully find nothing, but at least be looking in those areas that have the highest likelihood. It's a real challenging, as everybody knows, with state and federal budgets being so tight. It's really hard for different groups and agencies to add on additional monitoring. So we're hoping that by combining the data we've been collecting over the last year with some new information on life histories and species traits, we might be able to help refine that into some certain things to look for in certain locations.
Okay, another question related to that is you mentioned on one slide that certain, I think it was marine algae, have been identified, invasive species in California and Washington. Were those identified post-tsunami debris, or are those things that have just arrived sort of independently of tsunami debris?
Yeah, so those, let's see, I even have a couple more slides. So again, this is all information from Gail Hansen, but these have already been listed on other 100 worst lists or lists of worst invaders. So the Guadalupe is already on the 100 worst invader list in the Mediterranean, already known as the Red Menace in New England. So well known to be invasive in other locations, and also on the Japanese Tsunami Marine Debris, in this case in the reproductive state on one of these fishing boats I mentioned. The other one, the Undaria, commonly known as wakame, again is on the in the Global Invasive Species Database, already listed as one of the 100 world's worst invasive species. Quick growing, highly reproductive, really beautiful as well, but a nuisance fouling algae that's displaced native species along the west coast is already present in Southern California. So that was prior, already known about, but observed on the Agate beach dock. And the codium, which she has found on the tsunami, or the Olympic Peninsula dock, so the Mosquito Creek is the location closest to where that Washington dock is found. She didn't find that one reproductive, but it reproduces by fragmentation, and it's also on that 100 worst invaders list already, and already aggressively evaded North Atlantic. Dead man's fingers is one of the common names for that one. So all of those, at least the three big ones, and actually she hasn't listed those out yet. I know there's some species that haven't been reported on the west coast yet, but we also don't know if they're reproducing outside of tsunami marine debris yet either.
Okay, and we have one more. It looks like this may be our final question here. It says, has the barnacle on the macaw metal boom been positively identified to species, and is it related to JTMD?
I knew someone was going to ask me that, and that's probably someone from Washington. Jim Carlton, so a couple people have looked at it. Quickly look this up, and I know that there's two different possibilities on that one, and remember at the moment. If you, let's see, if that person or Jim wants to give me that person's name, or actually I could email Jim. Basically, I will look it up and find it because, yeah, it's not 100% definitively identified yet, but it's narrowly, it's very close. And yeah, I do believe it's not a species commonly found. So we're considering it likely Japanese marine debris.
Okay, thanks. I'm going to squeeze in one more question. It's just a little after 1 here, and we will make this the last one. Really appreciate your time today, Jessica. This question says, is there a protocol developed for sampling where docks or other objects have landed over the next couple of years to detect invasives? So that's, I think that gets back to the one you answered before about is anybody monitoring that stuff. But this specifically is asking, is there any protocol developed for that kind of sampling?
If you, I don't know if you can see this, I switched this over, but this is the blog I mentioned. So what we organized, if this is what you mean, right here is a collection protocol that we put together for the state folks that are helping us with sort of a data sheet to make it as easy as possible with some sort of ideal collection procedures on the second page, and then some idea of the kinds of things we're looking for and what we're seeing. I'm not sure if that's what this person meant, but that's out there, and we're happy to take feedback or provide more information if that's useful. And so anyone that's walking the beach or interested in trying to document it would go for, as specific location as possible, describe it, take some images, ideally all the way around, with known area like a pen or a quadrat or a ruler, estimate the percent cover, and then if you take any samples, just describe where they're from and what they are, and then people have been putting those in a box and shipping them to us or returning them to their state folks. So that's how we've been trying to coordinate with anybody interested in helping document what's out there. If you mean a more coordinated sort of regional response, again, that's what we're hoping to work on with Samchan and others, or EC Grant and around the region, to see if we can help translate the work we've been doing into some useful formats for the groups that could also help with more coordinated and widespread monitoring efforts for possible settlement or recruitment of these different species.
All right. Well, once again, thanks to everybody for their time and attending, and a huge thanks to you, Jessica, for your time presenting it. It's great to know about the blog, too, so that's a place where we can follow updates, because this is certainly an ongoing story. And this webinar will be recorded and archived on the Ocean Alaska Science and Learning Center website, and keep your eyes on your email for an announcement of next month's webinar, which will deal with integrating traditional ecological knowledge and Western science in a project that's coordinated between the Olympic Coast National Marine Sanctuary and the Seattle Aquarium. We look forward to that. All right, thanks again, everybody. Have a great day.